Hello and welcome to the Perilla Laboratories virtual poster presentation. My name is Alex Breyer. I'm a fourth-year graduate student and PhD candidate. Today I am pleased to provide an overview of the exciting and cutting-edge research performed by the Perilla Group. We are a computational biophysical chemistry research team focused on studying globally relevant pathogens with advanced theoretical models and the world's best supercomputers. Our models provide atomic resolution detail of complex viral systems, enlightening us to the physical properties of viral capsids, the protein cores of retroviruses that protect genetic material, viral lipid envelopes, and more. To any viewers interested in more information, or if you have questions about anything presented herein, please find Professor Perilla's email address at the top right of the poster and do not hesitate to reach out. Without further ado, let's begin. Among our principal research interests is elucidating the mechanisms of cellular processes relevant to HIV-1 maturation and infection. With atomically resolved models, we apply fundamental thermodynamic principles to analyze the energetic profiles of small molecule transport through the HIV-1 capsid and perform simulations that show what effects these molecules have on HIV-1 capsid stability. The simulations we perform are large in scale, describing tens of millions of atoms, often in physiological solvent environments, to provide the clearest glimpse into biology at the nanoscale. In addition to atomically resolved simulations, we employ coarse-grained molecular dynamics simulations of large systems, such as the HIV-1 lipid envelope, that allow us to investigate the physical properties of these systems over biologically relevant timescales. In addition to our work on HIV-1, we are broadly interested in viruses, elucidating their physical properties that aid our understanding and can inform downstream research such as drug development. For SARS-CoV-2, we are developing a complete multi-scale picture of the virion complex. Our model includes a lipid envelope encompassing millions of coarse-grained atoms, membrane M proteins, envelope E proteins, and spike S proteins, in addition to the nucleocapsid core containing the viral genome. Constructed in concert with experimental data, this complete model will provide us valuable information on the dynamical and structural biology of SARS-CoV-2, a virus which continues to affect the world a year after its initial outbreak. Similarly, we have performed cutting-edge work on the Ebola virus. Our atomistic simulations were analyzed to show the effect of the viral genome on Ebola's nucleocapsid stability. Further, we applied cutting-edge statistical analyses, such as principal component analysis, to the Ebola molecular dynamics trajectory, allowing us to reveal the system's essential dynamics. Our investigations into the physical properties and biology of viruses has culminated in many successful research projects, including studies of varying HIV-1 capsid morphologies, Rous sarcoma virus, chlamydia trachomatis, and more. The Perilla Laboratory is also invested in developing new computational biology methods that benefit both theoretical and experimental research communities. The various methods we have developed, and those in ongoing development, employ advanced theoretical and mathematical models, quantum chemistry, and the latest computing techniques, such as multiprocessor ray tracing, GPU computing, and artificial intelligence. One such example is the development of a high-performance tool that can rapidly analyze biomolecular systems and reveal properties such as the exchange rates of small molecules and ions, the transverse diffusion of lipids across a bilayer surface, and more, using parallelized ray tracing. Such a tool enables the analysis of terabytes of data in mere minutes. Our group also has significant interest in techniques to merge experimental data with theoretical models, allowing us to refine protein structures with nuclear magnetic resonance, or NMR, data, and cryo-electron microscopy, or cryo-EM, data. Further, we have developed AI which can even predict experimental observables such as chemical shifts from nothing more than atomic coordinate data. Other artificial intelligence projects include the automatic classification of viral particles from raw electron micrograph data, which intends to significantly expedite the time experimental researchers spend on data processing. One last example of a method under development in the lab is a tool that can analyze cryo-electron tomography data of viral capsids, namely HIV-1, and solve the coordinates of missing pieces of the capsid surface by applying fundamental mathematical concepts such as differential geometry and Fullerene geometry. To summarize today's short overview of the Perilla Laboratory's scientific interests and projects, we are a research group committed to advancing both fundamental viral biology and biochemistry, as well as the tools employed by the research community at large. Our lab is an exciting and stimulating place to perform research and advance our understanding of viruses and systems relevant to disease, as well as to work on the world's premier supercomputers and push the boundaries of high-performance computing. I hope you enjoyed this presentation. Please do not hesitate to contact us if you're interested or have questions about anything we discussed today. Thank you for your time, and I wish you all the best during your UD recruiting process.